that's a really good level of validation for us to show that actually your system, your mimicking system, behaves with already approved drugs in the same way that it does in human. That we're going to be looking at today. Oh, so there you go, CPU temperature. Is not my CPU temperature. It is something called organ-on-chip technology, or organs on chips. Now, this, I understand, will not be that familiar to a lot of people, and quite understandable. But if you're in the field of cell biology, or you're in the field of biology in general, you'll probably understand that there is something called model development, or that's ongoing, and models is the name that we give to experimental systems which are used to replicate what goes on either in the wild or in humans, most notably, and so they model what's going on, so they're called cellular models. And there is a consistent drive, especially in medicine development, and a lot in academia as well, as to try and develop systems and models that best represent what's actually going on in humans. And you know, we've started from very, very humble beginnings with this. You know, we've managed to culture cells in a dish, and you might think, okay, you got live cells, that's pretty good. They're likely to behave very similar to what goes on in the body. But of course, you're missing all the contextualization around those cells in that model. You know, you've got cells underwater in a dish or whatever, or under a growth medium in a dish. You know, they're not going to behave exactly like they would in the body, and we see that, manifested in its in things like clinical trials, for example, that has a whole load of what we call cellular assays, or cellular systems, or cellular models that, you know, drugs have gone through for years and years and years before they hit the clinical trial, and they've succeeded in every point in those cellular systems. They look fantastic. You put it into a human, put a new drug into a human, and nothing happens. And that happens more than it doesn't happen. I think the attrition rate is 90% in clinical trials, 80 to 90%, which effectively means that you fail. I mean, clinical trials fail 80% of the time, which isn't great. And the majority of times that they fail nowadays is due to a misunderstanding about what happens in the body versus what happened in a cellular system or even an animal system. And we can put that down to a number of things, but one of the main things you can put it down to is that the models that you're using, cellular models, are not representative of what's actually going on. Therefore, it worked in the cellular model, but it didn't work in the human. And so that's why there is a drive for new, innovative cellular models that represent human biology, in this example, a lot more than the ones we've already got. So where does organs on chips come in? So as you know, an organ is a system of interconnected cells, generally performing one task or a number of tasks, and there may be a number of different cell types, but they're all working together to perform some sort of function. The idea of an organ on a chip is to effectively remake that organ, or at least a part of an organ, in a dish, or in this case on a very small piece of plastic, and it's been nicknamed a chip. And there are loads of different types of organ on chip technology. It's very new. I think the technology, you could say the technology is probably around 5 to 10 years old, probably from its inception. I'm not sure when it actually started, but it's relatively new compared to other cellular culture mechanisms, systems that are decades old. So let me show you this. So what we're going to do, we're going to do the normal thing. We're going to do this on paper, and I've written out what we're going to go through. So we're going to talk about what organs on chips are, why do we need them. I've kind of gone over a little bit around that, but we'll go into the medical research standard animal research studies, cellular studies. Then we're going to talk about something a bit more in depth, which is validation of organ on chips, and I get into what that actually means, and then I give you some examples. And then we'll talk one step further and talk about organisms on chips, which is probably the next step. So let's go on to organ on chip. This whole talk has come about because I read this article on freethink.com called "Human-like organ chips could eliminate animal studies," and that's quite important, as we will find out. But you can see an example of this up here. So this is an example of a organ on chip physical design. This is someone's finger, so you get a idea of scale. No banana for scale, but this is someone's finger, so it will do. They're probably around 2 to 3 centimeters long, and they work on the nature that they can be this small because cells are quite small, and you can still put numerous cells in here. And a lot of these organ on chips, a lot of these chip structures, they have cells in them, but they also have what's called microfluidics in there. Now, as you're probably aware, you know, your body is awash with fluid on the inside. You have this thing called interstitial fluid, which is a fluid that effectively the cells sit in. You have solid organs, but they are actually, you know, they are not full fluids, if you like, but they do have an interstitial fluid that kind of runs through them. And you also have circulatory fluids, so like blood, and you have lymphatics as well. And these organ on chip systems aim to mimic components of this. So this is what it looks like, and this is just an image of someone at Harvard with an organ on a chip structure in an incubator, so you get a kind of an idea of feel for this. And this is, let's see if we can zoom in on this, get rid of this pop-up. What we have here is an organ on a chip connected to its microfluidics. So, you know, the cells will be in this layer somewhere, and these pipes will be pumping in either air or they'll be pumping in liquids, growth media, this type of thing. And interestingly, this whole chip is actually mounted on a microscope slide, because microscopy is one of the main techniques that are used by cell biologists to analyze and interrogate their cellular systems. And so what this group have done is they've made their chip amenable to imaging via light microscopy, and you can see it's just sat on a slide here, and I'm sure the focal depth will allow that to be imaged. But yeah, so that's kind of a cool overview. So let's just quickly start filling this in as we would normally do. So let's go to here. We are, what are organs on chips? So they are cellular systems. You're going to watch me misspell so much stuff, but fortunately Google has me covered with autocorrect. Cellular systems comprising multiple different cell types. So there you go. Can't type and talk. I still can't spell it. There we go. Look, I'm gonna have to go different. It's not difficult. It's different. Multiple different cell types, usually attached to microfluidics. And the aim is to mimic human physiology as best as possible. There are loads of caveats with this. You will not be able to mimic human physiology in a single chip. Not yet, anyway. We are too complex. Humans are too complex. We are, you know, arguably the most complex things in the universe. But we're getting there. And I'm going to bold this up because this is the ultimate kind of take home of this, right? Interestingly, this group is actually from the Harvard group. It's called it's either the Weiss or the Weiss or the Wyss Institute. Anyway, it's spelt W-I-S-S. Pronounce it however you like. These guys were the pioneers of organ-on-chip technology. I mean, they still kind of are. They're still developing it. Now it's a big industry. There's lots of different players within the industry. Other chips are available if you get that BBC reference. But we're going to show you how their chip works in a minute. And they've got a lung model chip, which is quite interesting, right? So why do we need organs on chip? We've explained what they are. Why do we need them? Okay, so in my field, which is kind of medicines development, as I mentioned before, you have to test and test and test and test and test and test new medicines, because you're potentially going to be taking a new chemical and you're going to be putting it into a person, and you're hoping it's going to be curing their ailment or their disease or whatever they're suffering from. But you are under no circumstances allowed to do anything dangerous, and this is where the rigor of preclinical and clinical testing comes in, and it really is rigorous. I mean, you may have heard the timelines of a medicine or a drug to be developed. It takes years to develop a new medicine, and I mean, I think the average time is about 10 to 15 years to develop a new medicine. Cancer drugs are a little bit quicker, but generally it's 10 to 15 years. And if you were going to divide that up into cycles, so what you would probably say is all of your preclinical work, so a lot of your cellular models, a lot of your animal models will be done in probably the first 5 to 6 years. That's a lot of time. With a new molecule, and then you've got that also involves finding the molecule. You've got to be able to discover it and develop it and make it better and stronger. But then you've probably got about 3 years of clinical testing, clinical trials to do, and then you'll have a few. You may have a regulatory year as well in order to kind of get it up to code in the regulatory space. But it takes an incredibly long time, and in order to make sure that medicines are safe, first of all they have to be safe, right? And then they have to be effective. And if you relate that back to clinical trials, you have a phase 1 clinical trial, which is a safety test. Is it safe in people? And then you have a phase 2 and a phase 3 clinical trial, which is it efficacious against your disease of interest that you're trying to target? But you have to make it safe, number one, absolute number one cardinal rule. And in order to do all the pre-work before it goes into a person, you have to have a lot of data on that molecule, and that data comes from cellular and animal models. And so medical research standard, I mean, you can literally just boil down this to test, 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 test again. Okay. Animal studies are called in vivo studies, and you have to provide a lot of information to the regulatory bodies, so the FDA, the EMA for Europe. You have to show toxicology. You have toxicology studies. You have to show what's called DMPK, which stands for drug metabolism and pharmacokinetics. And these are all done in animals, usually in rodents and then mammals, so higher mammals. So rodents and then a higher mammal. Usually the higher mammal could be a pig, or it could be a dog. Usually the rodents are rats and mice, as you probably would expect. And all of this is to predict human toxicity and human efficacy, right? And so the cellular studies do the same. So you might have a very, very initial kind of what we call a 2D culture, which would be a flat set of cells. So here you go. Like this is an example of what we would call 2D culture. And it's a little dish with some growth media on it, and underneath that growth media attached to the glass is likely to be a layer of cells. might look something like this. Okay, this is just cells growing in a 2D layer. They just grow as a flat sheet. But you can grow loads of different types of cells. You can grow, neurons. You can grow liver cells. You can grow epithelial cells that line the, tract of the lungs. But they are in fact just a 2D cell culture. So we have a 2D cell culture. You know what? I'll go through something that's a little bit more intricate, just because it adds a little bit of value. So in cell culture, you can generally split up cell culture into something called a cell line versus a primary cell. Now, we're still in the realm of 2D culture here. Okay, so we haven't gone 3D yet. We haven't gotten to the chip stage yet. We haven't done anything fancy. These are still just flat cells on a dish. But a cell line is effectively a cell that proliferates, divides over and over and over and over again. Usually it's what we would call a cancer cell line, so it's been derived from a tumor that's growing uncontrolled. And they are very, very useful in allowing us to have a piece of cellular material that we can just keep using over and over again. So you culture these cells, you do an experiment on them, then you've got more to culture, and you do another experiment. You can churn through these experiments very, very quickly, and they're very useful for that. The caveat with it is that these cells have been derived from, say, a cancer, or they've been what's called transformed in order to become immortalized. So normal cells don't grow forever. They grow for a bit, and then after a while they go through something called senescence, where they'll just stop growing. With cancer cell lines, they just carry on over and over and over again. But that means that they're inherently not behaving like normal cells in your body. Normal cells in your body do not proliferate over and over and over again. They will eventually stop. And so that is a difference. And if you're studying cancers, then that's fine, because you've got a nice model of the cancer and it's just growing and stuff. But if you're studying something, say cells that don't readily divide, like neurons, for example, say you're studying Alzheimer's, and you've got a neuronal cell line that's growing really, really quickly, is that actually going to be representative of what happens in your body, in your brain or your spinal cord or your nerves? It's unlikely. It's unlikely it's going to behave exactly as it would. It might behave to an extent what it's like, but it's not going to be exact. So the other side of the coin is something called primary cell culture. And this is where you actually culture cells that have come directly out of a body, like an organ of choice. So you can have a primary hepatocyte culture if you have a biopsy from someone's liver, you take the cells and you do an experiment on those cells in a dish. That's a primary cell culture. Those cells won't divide and divide and divide unless you immortalize them, and in that case you've changed them. So it's a trade-off. It's usability in cell lines versus things a bit closer to a patient and closer to a human in the primary cell culture. So immortal, and I guess, I don't like to use the term easy to culture, but they certainly are compared to the others. Primary cell culture is direct. We're on form with the spelling today. Directly from donor. Harder to culture. Usually they require more nuanced cultures, more specialized growth reagents that you have to give them, like growth medias and things. And not immortal, so you are time bound. Okay. So they are still your 2D cell culture. So there's something else that we haven't really mentioned, is 3D cell culture. Now this is relatively new, but fairly intuitive to kind of get your head around. 2D cells grow flat. They're not actually 2D, they're obviously 3D, but they're still very kind of flat. 3D cultures, they grow as more of a ball of cells, really. So you can imagine, this is a terrible image that I found online, but I kind of like it for its simplicity. they will grow in a 3D structure. Either they'll grow natively in a ball. So if you say you put cells into a dish, they'll lay out flat. But supposing you put cells into a round bottom flask, right? So that they'll naturally form a clump at the bottom. You can actually get cells to grow and proliferate in this environment. And some of them behave a little bit more, some cultures like that behave a little bit more like they do in a human body than flat grown cultures. What you can find is that they'll start secreting proteins and things like collagen, which will actually hold the ball together. And they've exemplified this as actually you can also have an artificial collagen structure that you can grow cells in. And I had a guest on this Monday night TwitchCast a few weeks ago called Naheem. He works in the realm of 3D bioprinting. So he prints using a 3D printer these kind of matrices, and then he injects the cells. Well, he prints them with the cells in there, and then they grow in whatever form you want them to grow in. It's really nice. It's a really innovative field, and you can print them into. You can effectively print cell cultures into structures of things like airways and say like gut epithelium and things like this. So you can actually you can include the 3D structure into your cell culture, which inherently will make cells a bit more cell cultures a bit more realistic. Okay, so 3D cultures provide very obviously a 3D framework, a pound 3 framework frame for growth. So you could say possibly, and I add the caveat here, possibly closer to human, or whatever animal you're looking at. I'm only using humans as an example here. Now, I will highlight the word possibly because we're going to talk about validation next after we've had a look at some organs on chips. Validation is important, and I will go through what that is. So, this is what I did. I didn't quite get to mention this. All right. So this is from the FDA. This is about animals in research, and the FDA, says that you effectively, for drugs, biologics, the focus of animal testing is on the drug's nature, chemistry, and effects pharmacologically, and its potential damage, toxicology. Animal testing is used to measure how much of the drug or biologic is absorbed into the blood, how much medical product is broken down chemically in the body, the toxicity of the product and its breakdown components, and how quickly the product is and its metabolites are excreted from the body. They need to know this. And so this is why you want to get it accurate, because if your animal study is not accurate, doesn't represent what's going on in a human, it will look great and you'll send it to the FDA and the FDA will approve it and your clinical trial will fail, and that will not be good. For anyone, that could be 10, 15 years work down the shitter. Official terminology, that. And the other thing to mention around animal usage is in the UK, at least, and this has kind of been adopted pretty much worldwide, is actually we have something called the NC3Rs, which is a charitable body. I think they're headquartered in London. It stands for the National Centre for the Replacement, Refinement and Reductions of Animals in Research. And pretty much every academic lab and certainly every big pharma company adheres to the 3Rs. So everyone is trying to actually reduce, refine or replace animals in their studies. Because, one thing, because of the ethics of it, you don't want to overuse animals. You don't want to use animals where you don't have to. And secondly, you want things to be as translatable as possible. And we know that a mouse is not a human. In fact, ironically, we probably know more about mouse physiology than we do about human physiology right now, because so much testing has been done. But the majority of people will abide by the 3Rs, and they will be looking for alternative strategies. Hence why we end up with organs on chips. So I didn't mention that in this animal study, but I will literally just put NC3Rs